Now let us examine in detail about flower color in Lathyrus odoratus. Betson and Punnett in 1904 studied the inheritance of flower color in sweet pea Lathyrus odoratus. Sweet pea is an annual climbing flowering plant to a height of 1 to 2 meters, where suitable support is available. It belongs to the genus Lathyrus and the family Fabaceae. The plant is native to southern Italy, Sicily and the Aegean islands. They opted to use sweet pea plants because it is relatively easy to perform hybrid crosses with these plants and chose to focus on the characteristics of flower color. There are two varieties of pea plant, one producing red color and the other producing white color. They selected two white flowered true breeding varieties of sweet pea plants with genotypes C, small a and small c, capital A respectively for the study of inheritance pattern in sweet pea. When two true breeding pure line varieties of white colored sweet pea plants with genotypes C, small a and small c, capital A were crossed, the F1 progenies produced red flowered plant with the genotype C, capital A, small a. In F2, the F1 plants were self-pollinated among themselves. In F2 generation, the offsprings resulted in the ratio 9:7, of which 9 were red, 7 were white. In F2 generation, out of the 16 progenies, 9 showed phenotypically red flower and 7 appeared as white flower. From the above experiment, it is to be noted that red flowered sweet pea plants with both complementary C and A genes were in 9 progenies, whereas white flowered sweet pea plants in which either of the complementary gene C or A is present are 7 in number. After many years, Bateson first described this modified 9:7 phenotypic ratio in sweet pea plants. Researchers were finally able to determine the two genes C and A responsible for it. The surprising results can be analyzed by understanding the mechanism of color production in flowers. The table shows the 9:7 ratio is a modification of not genotypic but phenotypic Mendelian ratios. Note that the C and A genes are sought independently, and note that the presence of a recessive genotype at one locus, that is small c or small a mask the effects of the alleles at the other locus. It is obvious that both the dominant alleles C and A is necessary for the production of pigment in flowers. There are 7 combinations of either a small c or small a which would yield the white flower phenotype. Hence, the 9:7 phenotypic ratio of purple to white flowers. The cross can be represented as follows. The above experiment showed that the red colored flower is due to the interaction of two complementary genes C and A. But the white colored flower is lacking either C or A genes in the genotype of the offsprings. In the F2 generation, the pea plants with both C and A genes are present together in the genotype, which produces the red coloration due to the phenotypic expression of dominant gene A along with the complementary C gene. The interesting progenies in the F1 and F2 generation were analyzed for the mechanism of color production in pea plants. These genes, capital A and capital C, control sweet pea flower color by controlling pea plant biochemistry, in particular that related to pigment compounds called anthocyanins. The genetic analysis showed that the color in flowers is due to the action of the pigment anthocyanin. In peas, there is a two-step chemical reaction for the formation of anthocyanins. Gene C is responsible for the first step and gene A is responsible for the second. If any step is non-functional, there is no purple pigment is produced and the affected sweet pea plant bears only white flowers. The dominant capital C and capital A alleles code for functional steps in anthocyanin production, whereas the recessive small c and small a alleles code for non-functional steps. Thus, white flowers will result if two recessive alleles occur for either gene. Here, the capital C gene is responsible for the production of the enzyme that catalyzes the necessary chemicals for production of an intermediate complex in anthocyanin pigment formation, whereas gene capital A is responsible for production of enzyme which catalyzes the intermediate colorless complex into anthocyanin, the color producing pigment. Therefore, both capital C gene and capital A gene have their combined effect in production of anthocyanin pigment, but alone capital C gene or capital A gene cannot help in production of anthocyanin pigment.